introduction. Welcome everyone to this webinar session. As the title tells you, I will be talking mostly about single molecule design with a tool that is relatively new in the ChemAxon portfolio. And what is special about it is that we hope that it would solve the hassle that usually comes with working with other chemists in single molecule design projects. The typical drug discovery workflow can be, to some extent, described with this sort of iterative cycle that you see here in this slide. So you start somewhere with evaluating data, extracting chemical information and other metadata from publications, patents, whatsoever. And based on that information, you start some sort of molecular design. Those molecules that you have designed are then evaluated in one way or another, and the interesting ones are then registered to a database. And that's where it starts over again. You reanalyze data, you add new information to that, and start the design cycle from the beginning. Now, what we will be focusing on today starts with extracting chemical information from scientific publication. Then we will continue with the design step that will involve multiple scientists, well, in this case only two, but it could involve any number of scientists using the tool that I will be showing to you. And then at the end of the design process, the interesting molecules will be registered. Everything that I will be showing to you will really showcase the functionality, so please do not expect any real scientific information from this webinar. Now, if you want to simplify this workflow, then you look at something like this. You start with some sort of information that comes from publications, patents, or in-house libraries, which can be real or virtual libraries. You collect this information somehow, and then you start exploring this chemical space. And at some point, you start also expanding this chemical space and continue with the analysis and see where you can go from that starting point. Now, with enough information, you can refine and optimize the molecule that you are working on. And at some point, you can come to consensus and say that, okay, this is the direction where I want to go. And if you get to that point, you also want to register that information in a registration database. So where we start with is really how you can get the starting point and information from various documents. But in order to explain that, I have to start from the basics. I'm sure that most of you are aware of ChemAxon's naming suite, which is capable of converting chemical structures to chemical names of all sorts. IUPAC names, common names, and other chemical strings as well. And it works also vice versa. But in many cases, it is not that simple. The chemical information occurs in every possible form in various types of documents. It can be internal IDs, CAS numbers, all sorts of chemical strings, all the objects as well in some cases. And also the documents can be very versatile. So what we have designed to be able to extract all this chemical information from various documents is document structure. These documents can be, of course, of various types, scientific publications, patent documents most typically, but also web pages, internal reports, and so on. Now, document structure works with a collection of documents. You can extract the chemical information from those, collect them into chemical files, but maybe you want to step one level higher even from this point. And you want to do something like extracting the information from these documents and importing them directly into a database so that you can make this information searchable for everybody. And you want to make this information retraceable for everybody. So what you want to achieve really is to be able to maintain a repository of documents where you can just type in a name of a molecule or draw a chemical structure, and based on this chemical information you want to be able to search in your document repository. And this is what document to database does. So it allows you to mine in all the documents that contain relevant information for your projects and do this using a database backend. Now, on top of document to database, what we have built is a very user-friendly web interface, a web client basically, which we call Plexus mining, which allows scientists to easily search and browse in these document repositories, ask chemical questions in the system and retrieve the answers immediately. And it speaks actually many languages, not just English, but also Japanese and Chinese, as you can probably see in this screenshot. Now, document to database is, as you I'm sure understand by now, designed to be high throughput. It is fully automated. It scans through large document repositories, extracts the chemistry in an automated fashion, and synchronizes the database with new coming information immediately. So it is really designed to be a fully automated system. And it also means that in this case no real manual curation is involved at any level. Now, this also means, on the other hand, that in some cases some inaccuracies may occur. If you want to go really into the details, and especially if you want to extract, for instance, high accuracy Markush structures from documents, which is extremely relevant for maintaining patent databases, for instance, then you might want to use a sort of a different tool. And we call this tool ChemCurator, which is presented in ChemAxon's portfolio. And in contrast to document to database, ChemCurator does require manual intervention at some extent. But in exchange for this extra effort, it provides you with very high accuracy. In contrast to document to database, ChemCurator typically processes one or just a few documents at a time. And using ChemCurator, you would be able to extract very accurate Markush structures from patent documents. But it is also very widely used for extracting chemical information from scientific publications. Now, from this point on, I will leave document to database and document to structure technology, or to be more precise, document to database technology behind, and I will be focusing more on ChemCurator. So what we see here is ChemCurator's interface. This is really the user interface that a user would see. On the left-hand side, you can see the project explorer, and there are now 3 projects open in the screenshot. In the middle, you see the annotated document, and the highlights are referring to the structures that have been located in the document. On the right-hand side, you see a list of compounds that have been exported from the document. So these are structures that have been recognized to be mentioned in the document, and that the corresponding structures have been generated on the fly. And in the bottom, you see a collection of selected structures. Now, there's another view for ChemCurator, and this is especially relevant for patent documents, as I mentioned before. In the middle, just like before, you see the annotated documents, but on the right-hand side, you see a Markush editor where you can edit and curate the Markush structure that has been exported from the document. On the left-hand side, you see a structure checker window where any errors that occur in the structures will be shown and can be automatically fixed. And in the bottom, just like before, you see the selected structures that you want to work with. You may have noticed that on the right-hand side in the Markush editor, there are some exemplified structures. And some of them are highlighted in green, others are highlighted in red. This is indicating whether the exemplified structures are actually matching the Markush structure that you are working with. So this is a good indicator on how far ahead you are with curating your Markush structure. Okay, so if we get back to our workflow, collecting this information from the publications is typically, well, a single-man job. But the rest of the workflow, where you're exploring the chemical space and expanding that space, and going any further with analysis and then getting to the optimization step, involves more people. This is where collaboration becomes really important. And well, getting to a consensus sometimes requires even more people to be involved. Now, this means that what you need to proceed are really good meetings. And a good meeting typically involves the right people. It includes all the data that is needed to make good decisions. The data needs to be up to date and needs to be easily available as a matter of fact. And preparations for these meetings can be really difficult. You also need to be able, during the meeting, to react to changes, to react to information that just pop out during the meeting. And at the end of it, you really want to create a report that is reflecting what has happened in that meeting. Now, as you all probably may have experienced already, it is not how a typical meeting goes down. Especially if you have research partners who are not sitting in the same office as you are, there can be all sorts of problems. First of all, with the scheduling, then with sharing the appropriate information with people. You might end up distributing PowerPoint files that get outdated by the time the meeting actually starts. It is really difficult to get to the relevant information on the fly and so on. So it can be a real pain. And also, of course, when you want to generate a report, capturing all the information that has been mentioned during the meeting can be really difficult. So what we believe can be a solution for all these difficulties is Marvin Live. When we designed and developed Marvin Live, the goal was to allow people to work with off-site colleagues, well, basically focusing on small molecules, and do it in a very easy and slick fashion. We wanted to allow people to do this with a minimal IT overhead and a very minimal learning curve. What we are trying to provide here is for researchers to get relevant information during the meetings on the fly so that discussion stays on the right track. And also to simplify, therefore, not just the meeting preparation, but also the follow-up by generating automated reports. In the heart of the application sits a shared editor. What you see in this small animated GIF is basically two chemists sitting in two different locations. On the left-hand side, as Dave is editing this molecule, Susan, sitting in a different office, sees the notifications instantly, and vice versa. And actually, any modification that is done by one chemist is also highlighted in the other chemist's Marvin JS canvas by the user core that is assigned to that particular chemist who is doing the edit. So this is a fast and self-updating shared editor that allows people to work on the same molecule at the same time. Now, besides the molecule, what Marvin Live also provides is immediate feedback on the molecules that people are designing. And this feedback comes from various sources. On the one hand, it can come from database cross-checks, which send the molecule under discussion as a query structure to those databases and retrieves the relevant information from those databases. On the other hand, it can also be applying various models on the molecule that you are designing. These can be simple physico-chemical calculations or property predictions, but also more complex models like, for instance, a 3D alignment, like in this picture. Now, mining the data that is relevant for your design can be of various types. In this particular example, what I'm showing is that, for instance, getting that information from an assay database can be really important for a meeting. You see which way you want to go, whether there is information available of the molecule that you are working on or not. You can even look at the e-molecules database to see what would be the easiest way to purchase their own building blocks when you want to synthesize a molecule, and so on. On the other hand, you also, like I said before, want to explore the actual property space of the molecule to guide the design in the right direction. In this example, there is a reference molecule against which a three-dimensional alignment is done, and that helps chemists understand how that particular molecule would overlay on the reference structure. I will tell you more about it in the actual demo part. But in the bottom of this screenshot, you see how various calculated properties are actually changing on any modification against the reference structure. These are indicated by these small arrows. Now, once the design is done, or at least reaches some stage, what you want to do is to capture the information from the meeting. And this is what the automated report generation plugin is there for in Marvin Live. It will save you the relevant structure or the important structures into various document formats. It can be a PowerPoint or an MS Word document for reporting, but it can also be a molecular file like an SD file or a Marvin document if you want to upload them to a database, for instance. Or you can even save the molecules that you have designed directly to a compound registration system. This is a customizable interface, just like any other plugin that is available in Marvin Live. So basically, you can plug here in anything that you desire. Just a few words about security. This is a web application, so information is exchanged over the web. Therefore, it requires the appropriate security measures. So we do apply standard authentication protocols that are, as you can see, listed here. You can create private rooms for yourself if you like, where you can do just your own design and then share those ideas with others later when you feel that you are mature enough. The rooms do not require any scheduling. You can just create a room and invite anybody to share the discussion with you, as you will see in a few minutes in the demonstration part. Now, one typical use case for Marvin Live would be, well, maybe the lowest scale use case when a chemist, biochemist, computational chemist, or biologist is using the application alone. So he or she would just design the molecules or analyze the molecules that they have imported into the application, save their ideas in an idea repository. So it can be thought of as a private playground. But then at some stage, they can invite their colleagues to share the discussion. These can be internal colleagues in different locations, and they can, like you have seen before and we'll see later again, they can be working on the same molecules and share their ideas with each other. But the collaborators can be also external, so it can be CRO partners or academic collaborators. So really, the application can be used from very small scale up to really large scale use cases with multiple collaborators. Marvin Live maintains various domains, so if you don't want CRO1 to see what CRO2 is discussing with you, the information can be, of course, separated from each other in a separate way. Now, once you're done with the design, what you want to do is to save the interesting molecules into your compound registration system. And this is actually the last step of the workflow. So let me just tell you a few words about our compound registration system. The most basic workflow, how the actual registration process can be described is as follows. You submit your structures. There is some sort of a validation and normalization process where salts and solvates are recognized and the tautomers are handled. The system also checks whether that particular structure that you are submitting is already present in the database or not. And then based on this information, it either generates an absolutely new ID for your structure, or it registers it in the appropriate place in the hierarchy. I will tell you more about the hierarchy in a few seconds. So this compound registration, as one of my colleagues, Markush, used this analogy just a few days ago, and I really like this analogy, can be thought of in a similar fashion as the air flight. So you book your ticket, is the analogy for submitting your compound in whatever format you have generated it. And in the next step, when you are checking in, it is handled to be stripped from its salts and solvates and getting standardized. And there is also a security check step where the system makes sure that everything is okay with your structure. There are no mistakes that you would save in your database. And once everything is fine, the structure is actually loaded into the database, which is sort of equivalent to you boarding the plane and starting your journey. Now, this is how the user of the compound registration system would see it. What we do in the background is a little bit more complex. I really don't want to go into details with this. I just want to show you this workflow very briefly so that you maybe understand more deeply what is actually going on. So the submitted data is validated and normalized in multiple steps, and errors are checked for and corrected on the fly if possible. If at any stage of the normalization or the error checking or even the duplicate matching the process fails, the submitted data, the structures will end up in the staging area where they can be revised and the appropriate action can be made. If it goes through the system, it will be registered at some level of the hierarchy. I have mentioned hierarchy twice already. So what is this hierarchy that we are applying? We're storing the structures in the compound registration in a hierarchical fashion. On the top of the hierarchy sits the parent structure, which is really a virtual representation of the molecule. This is salt-free and isotope-free, really just the pure structure itself. To each parent structure, multiple versions can belong. A version can be, for instance, one or another salt form of the particular molecule, or an isotopically labeled version of the molecule. And then to each version, you can register various batches. So when you are doing your duplicate checking, the system will see at which level the structure that you are submitting is already present and will create the record accordingly. The structure matches are taking into consideration not just the topology of the structure, but also, of course, additional features like the tautomer information, the stereocenters, and so on. But also additional metadata can be taken into consideration, like chemically significant text. If a match is found in the system will ask you what to do. Well, it depends on the configuration, of course. But what typically happens, that the registrar would be able to decide whether they want to register a new record for the particular molecule, accept the existing one, or replace it. This is just some minor technical detail. Alright, so this was the presentation, and let me jump to the actual demonstration part. So what I will start with is ChemCurator. What I have done in advance is I have loaded a PDF file, a scientific publication, that I want to very briefly analyze. I have reloaded this because it takes approximately a minute or so to parse the document, and I didn't want to waste time. So what you see here is really just the document itself. On the left-hand side, as you remember, is the project explorer. There are a few other projects already open in this application. But here in the middle, you see a document that belongs to this ChemAxon article. You may have noticed that there are a few highlights in the text. So if I click on these, you will see that the corresponding structures have been already generated. But they are not available in the collection of structures. So what I will do now is I will add a new compounds collection to this project. And I will just ask ChemCurator to extract the molecules. And I will not apply any filtering. So as you can see, this was pretty instant, and 179 molecules have been extracted from the document. There is a few additional information that comes with the molecules, like names, for instance, that have been found in the document. Now, what you can do if you don't want to export everything is to apply filtering on the fly when you are doing the export. So in this case, I will apply a substructure search while exporting molecules from the document. And I will just use naphthalene for as a query structure for substructure search. And what I will get is just 6 molecules in contrast to the 179 that I had before. As I go through these molecules, you will see where they are located in this document, either as a name or even images are converted to chemical structures. So what I will do now is I will export these structures to my hard drive. Structures. So there they are. And what I will do as a next step is I will create a meeting room in Marvin Live, upload these structures, or at least some of these structures, and I will invite one of my colleagues to join me for a short session in Marvin Live. So I go to my browser. This is where I can select which domain I want to log in. I will select the internal domain, and I will be prompted to type in my username and password. So I'm logging into this domain. There are already a few meetings that I have created earlier. One thing that you might think is important about Marvin Live is that it supports persistent meetings. So a meeting can run basically forever, unless it's configured otherwise. So you can revisit meetings later on and see what you have discussed with others a few weeks or a few months ago. This is, I think, a really useful feature. So I will just add my name that will appear in the meeting, and I will create a new meeting. And I call this design November 11th. I could create a private meeting, which would be only visible to me, or a public one, which would be visible to everyone who has access to the domain that I'm logged into. So in this case, I will create a public meeting. And what I will do as a next step is I will invite my colleague, Markush, to join me in this meeting. All I need to do is copy and send this URL to him, which I have done. So in a few seconds, Markush will join the meeting. In the meantime, I'm uploading the structures that I have just exported. this is the dialog that appears when I'm uploading structures. As you remember, I have exported six molecules from the document, but I don't want to upload all of them. I select two that I want to upload, and maybe these two fields that came with them, I will also add to the meeting. So I just click on Add To. So you see these two molecules appear right here. In the meantime, Markush has joined the meeting. So I just say hello in the chat area. Okay. This is what I want to discuss with you. I'm working on these structures because what I want to do is explore some naproxen analogs for a Cox project that we are working on right now. And what I want Markush to do is to help me with the design. So what I will do first is explore possible metabolic site of naproxen itself. And in order to do that, I will load the reference structure for the cytochrome P450 molecule that has been co-crystallized with the molecule, because what I will do is I will do this co-crystallized structure as a reference for 3D alignment. So there are two ways to do that. Either I could just use the alignment itself in 3 dimensions, and you will see that if I load naproxen itself into the editor, I will get the alignment right away. But this doesn't tell me too much about potential metabolic sites. So what I want to do instead is to load the protein and see how the alignment looks in the proximity of the hand. And maybe you can't see it very well here, so I will just put it full screen and show it to you. So what I see now is that actually this methoxysite is very close to the iron. I can measure its distance. It's 4.38 angstrom, so it really seems like a sensitive site from a metabolic point of view, which is not very good. So I'm not happy about it. So I suggest to replace this methoxysite with the chlorine. But before I do that, I want to save this as the next reference, because I know that any modifications that I will make will also modify the property space of the molecule. So I pin naproxen as my next reference structure. I can close this window now, and instead I will open a window for the calculated properties. So these are just a few selected calculations that are done on the molecule, but any modification I make from now on will be reflected in these calculated properties. So I just replace this with a chlorine. And you will see that the C log P has been increased. So I think that's something that worries Markush. And indeed, yeah, that's true. So as you can see here in the chat area, he's worried about a high C log P that can be problematic with a high plasma protein binding. So we should really decrease it somehow. And he has an idea. So now he's going to edit the structure. And as you will see, I will get this indicator that he is now editing. I see also the highlights. I see the atoms that he is adding to the molecule. And I can also see instantly that the log P is decreasing. So we are really happy about it. Like I mentioned in the beginning, we are not taking the full design process here. It's really just to showcase some of the functionalities. So what I will do now is, besides the properties, I want to see whether this particular molecule has been already registered to a compound registration system. There is no matching data, which is good news. There are a few other database checks that I could do, for instance. I could check the assay database to see if there is anything relevant for this particular molecule. But probably there wouldn't be anything because it's not even in our compound registration. So I say that, okay, this looks interesting to me, so I make a snapshot of it. Here I can add some action item. I could say that relevant for Props project, which is not exactly an action item, but I could say something like, Markush, please check potential synthetic pathways by Friday, or something like that. And now I will remove this one for now, but I will save this as a snapshot. As you can see, this structure is marked by Markush's color because he was the last one to edit this molecule. Markush's color is blue, as you can see here at the bottom. Okay, so this is fine. We have come to some consensus with this design. So what I will do now is to save this structure to our compound registration system. The request has been sent, and I can actually track my structure in compound registration. So I just need to log in. And there it is. So this record has been processed. This particular user that I have used for the login has submitted 88 structures in total. 85 of them were successful, and there were a few that failed. I might need to check out why they have failed. There was probably something wrong with the structures. But this particular one went through, so I can actually take a look at it in the system. So here it is. And you can see how this is matching data hierarchy that I was just trying to use. So the parent ID, it got what is ChemAxon72. The version identifier is ChemAxon72A, and one batch has been also registered to it. If I go back here, I'll just click OK. I could even save other types of reports. So for instance, Microsoft PowerPoint report would be relevant. It's downloading all the structures that have been discussed during the meeting. And relevant properties are also added to the molecules that were calculated during the meeting. Okay, so what I will do next is I will just say bye to Markush. This was cool. And this pretty much concludes the demonstration part. I hope that everything was clear. And now I hand it back to Jan.